काय करतोय? चार्जिंग करतोय?
नाही रे, माझं हे लावलं कॅमेराचं नवीन हे टाकली ना.
हां
गॅस मला ना म्हणजे टाकायसाठी.
हां हे काय?
ते तो पॉइंटर असतो ना, लेझर. तुम्ही तेच सर्व्हिसला आहे ना?
हे असू शकेल. हे गरम होईल. हे बघ, हे गरम हवा येते.
तुम्ही काय म्हणाले? अच्छा.
One session, and I always hate to be the first speaker after lunch, and I was glad when the vendor slotted me before lunch, but it was not to be. So I have the difficult task of not only keeping you awake, but also to keep myself awake. And fortunately, we have the first dose of Viagra in the morning, and I guess all of us, along with the food, probably will have a long-term effect on keeping us awake through the afternoon session. I think India has done well in the last millennium in terms of the process patent regime, in terms of our self-sufficiency in pharmaceutical business, and also in achieving balance of payments in the positive note like the information technology. But I don't think we have done enough in the global context when we are going to see in the new millennium the IPR rights, patents and liberalization coming through, where India will be in the future. And there's a lot of discussion going on in terms of whether we should continue our efforts in our bulk drug exports to both the developing and developed markets, whether we should focus into the generic business of the world, whether we should focus into drug delivery systems or into new chemical entities. And as you know, most companies in India don't have the funds to do new chemical entity research. I believe more and more companies should and could focus, and we have the capability to focus into drug delivery systems. And as Ajit Singh mentioned, there are examples of success stories, and I'm sure there will be many more in the future. So what I'm going to spend time on today is business, technology, and regulatory compulsions of oral control release systems. I think this presentation, the theme was presented sometime in '98 in one of the Oral Control Release Society meetings in Mumbai. And I think today it's much more relevant in view of the IPR regime coming in place as to where India should be. And I always ask the 4 W's, the what, when, why, and where we should do this kind of research. Next slide. If you look at the global scenario, there is the breakup of the United States, Europe, and Japan states the major piece of the cake, and India, as you see, is a minuscule of that total global market. In terms of the total share of the business, you can see that 80% of the total market is taken by new chemical entities. However, there is about 10% market share, both in terms of generics and drug delivery systems. There is a belief that in the new decade, generics will probably increase to around 20% or more, and NDDS probably will continue to remain at around 10 to 20%. Now that I see as an opportunity that Indian industry can certainly tap, along with the innovation which many of the companies like Rantaxi and Reddy's are going in for new chemical entities. In terms of the strategy, cost, and time required for new chemical entities and drug delivery, it becomes very obvious that most companies cannot invest money in new chemical entities which costs something in the region of $300 to $400 million, and it takes almost 10 to 15 years to take a new molecule from conception to commercialization. And of course, the clinical research, which constitutes almost 50 to 70% of the total cost. Whereas if you compare that with generics for drug delivery or innovative drug delivery systems, this can be done within a span of 2 to 8 years. Average cost would be anything from $2 million to $30 million, and the success rate certainly is much greater than new chemical entities. You can choose either to be a generic drug delivery systems where an innovator exists in the marketplace, or you can have an innovative idea converted into NDA route through the clinical research program for a new drug discovery, new drug delivery systems. So you have in NDDS either an NDA or an ANDA situation. Next slide. NDDS is a big word which has a number of categories of products and technologies that are currently being worked on. And some of them are the oral control release, inhalation or transmucosal systems, implants, transdermals, and there are many other smaller areas of drug delivery. 2-3 things are very apparent here. One, that the oral control release takes the major piece of the cake of the total NDDS market, which is almost 70% of the total business. Apart from that, the other areas which are making big inroads are inhalation, implants, and transdermals. I believe that most of the implants, transdermal, and inhalation technology is still at its infancy in terms of controlled drug delivery systems, and most of the work is going on either in drug delivery companies or majority of the academic institutions. They're still at the infancy stage, whereas the controlled release systems have been developed into commercial successes. It is well understood with the advent of the polymer technology and the equipment upgradation that has taken place globally and also in India over the last decade. And therefore I believe that oral control release is going to continue to remain our focus area. That is going to show the biggest growth area in the short term, followed by probably transmucosal and transdermal taking over towards the later part of the next decade. If we concentrate on oral control release, this is a classical curve that I show in many of my presentations to highlight a number of issues on this slide. One, that you see most of the products are given in a frequency of dosing is high, and therefore you see a number of peaks and troughs. The peaks results in your toxicity or the side effects, and the troughs gives you the non-compliance. And probably in the 70s and 80s, if you had asked the question, could I give this product once a day, where we have the steady state blood levels within the therapeutic window, and you avoid the side effects of the peaks and the non-compliance of the troughs, and maintain a steady state which beyond that also offers the patient a better convenience and better compliance in terms of the system. However, there are two options. Either as an innovator, you have to maintain that steady state, avoiding the frequency of dosing, or in case that there is a product which is existing in the market that you're trying to match the profile of the innovator. Often, a generic drug delivery systems is difficult to make because you have the profile to match throughout the 12 to 24-hour period. And therefore often, although cost and time-wise it might take less, but often it can be a very daunting task. This is an example of how one can extend the product cycle by offering the patient convenience and the compliance that I mentioned earlier. And this is a classic example of Cardizem. Diltiazem was the generic product, where you can see this product was introduced sometime in the early 80s, and it peaked to around $500 million around the mid-80s. With the advent of the ACE inhibitors, this product profile started to decay with products like Captopril and Enalapril as a once a day, and therefore you can see the declining phase of this drug. However, in the meantime, Elan developed a controlled release product starting with the SR, which was a twice daily product introduced sometime in '89. But very quickly, they were able to develop a technology with the help of some newer polymers and newer technology into a once-a-day beats technology, which they introduced as cardiac CD sometime in early 90s. And two things become very apparent. One, that it had a new lease of life in the early 90s. Today it enjoys almost $800 million sales in the United States and combined sales of almost $1 billion worldwide. So it enjoyed, one, a bigger market share of the same molecule, mainly because it offered better patient convenience and compliance. And even to this day, there are very little generic competition because of the difficulty in matching the blood levels of an existing once-a-day profile. I understand now there are companies like Andrex who have achieved that bioequivalence, and they will be coming under the generic threat of drug delivery systems. However, it has given a new lease of life and an extension of the patent, and the other problem is that you have to break the technology and come up with your own patentable process in order to have a bioequivalent generic product. And there are many more examples like this. For example, PRODUC also had the same kind of profile after nifedipine came under the generic threat and became a patent expired molecule. Next slide. The two options that we mentioned, either you can become the NDA if you have a new idea for an existing molecule to make it low frequency dosing and a better patient compliance. You can be the innovator, in which case you'll be going to the new drug application. Or in the case of an existing product, if you want to be a generic substitute, you can go to the ANDA route. Now the two strategies are quite different. In the case of being an innovator, you'll be going to the clinical option, and therefore the cost becomes much higher, in the region of $20 to $30 million, and the development time is longer because of the long clinical period that the product has to go through. However, the success rate is higher than a generic product because you are the innovator, where you're creating your own once-a-day profile based on your technology or the polymer system that you wish to use. Whereas the ANDA route is cheaper and it takes less time, and many of the Indian companies can go through a generic substitution route to get into the global market. However, it becomes difficult because you have to match the profile of the existing molecule. And the couple of examples that you have later on gives you a very good example why there are not many generic products of the same drug delivery systems that have been there for the last decade or so. For example, PRODUC was introduced as a once-a-day, the first pH-independent matrix system developed by ALZA, the famous Oros technology with a laser drill, which was marketed successfully by Pfizer. Then we have, of course, Cardizem CD, which was also developed by Elan as a beads coating technology, the first extended release once-a-day product. And there have been a number of other products which have probably been attempted as a generic substitute to Cardizem CD, but probably in the absence of matching bioequivalence profile and a good once-a-day profile, Yago has developed geometrics technology to have a once-a-day diltiazem, which was licensed to Watson as a Delacor XR, which is currently in the market, and subsequently also BioVale introduced another once-a-day. Each one of them are fairly big products for such an old molecule because of the better advantages that it offers to the patient, the consumer at large. Next one. In terms of the critical activities, the milestones that one has to develop, either for a generic or an innovator, whether you go through an ANDA or ANDA route. In the ANDA route, you have to do all the typical examples of formulation development to develop your prototype formulation with a therapeutic window, pharmacokinetic profiles. Then you have to establish your bioequivalence studies, the pilot bio studies and the pilot stability studies, followed by your pivotal activity, which is your exhibit batch, stability and bioequivalence studies, followed by your ANDA filing. Time scale required roughly is typically around 2 to 3 years. Whereas if you are the innovator going through the clinical route, apart from the above studies that you will do, you will require to do the clinical studies. It might require, depending on the molecule, the preclinical safety and toxicity, followed by your number of the phases of the clinical program, depending on the product, depending on the drug that you have in mind. In this case, the time scale is much longer, something in the region of 5 to 8 years. I just wanted to touch upon the regulatory aspects of drug delivery systems. Again, it's no different from the generic strategy that United States FDA had introduced in 1984, which is well known by many of us as the Waxman-Hatch Bill. It was introduced essentially to avoid the expensive cost of introducing a molecule even for the second and third time, where the innovator has to still go through the clinical program. And they came up with this innovative idea that if the innovator exists for the same molecule, for the same potency, for the same dosage form, you don't have to do the clinical studies, which constitutes 50 to 70% of the research cost. And therefore, you can cut down on the cost and time and reduce the healthcare cost by introducing a cheaper generic substitution of an innovative product. This obviously led to an explosion of generics in the 80s and 90s, and we have seen some examples of generic drug delivery systems which have also started to appear in the short term. And I think what we'll see in the future, there will be a large portfolio of innovative drug delivery systems going through the NDA route, as well as there will be generic substitutions of those drug delivery systems as and when the product and process patent expires. In terms of the regulatory guidelines, the two major guidelines, which is European and the United States, I just wanted to bring in the perception of what is required for controlled release systems. Unlike for generic products, where you would only require to do the bioequivalent studies on the highest potency and you get waiver on the lower potencies, in the case of drug delivery systems, you would be required to do testing on every potency of if you have a multiple potency products. However, there are differences in terms of the dosage forms. If you are making a tablet, you would not only require the single dose fed and the multiple dose studies, but you have to do the single dose study for each and every strength of your drug delivery systems. However, you will get waiver for the low potencies in terms of the multiple dose and the fed study. In the case of capsules, however, you don't have to do the single dose study for all the potencies. You will only require to do it for the highest potency. At the same time, you continue to do the high potency bioequivalent studies, both for the multiple dose and the fed. The reason you get the waiver for the low potencies in the case of capsules, because of the assumption that the scaled down formulations of your capsule product will require lower proportions of the fill weight of the same technology and the same beads, and therefore you get a waiver on the fasted study of the low potency product. In the case of tablets, while you scale down your formulation, often your dissolution, your blood levels changes because of the size of the matrix that you're filling in the case of tablets. And therefore, there have been cases where your blood levels may change as the size of the tablet reduces due to the potency. And therefore, FDA and MCA insist that you do bioequivalent studies for all potencies. Often it might be prudent for a development scientist to decide whether he wants to do a tablet or a capsules, because the cost of the studies often can be quite significant depending on the dosing form that you're developing. Next one. In terms of the guidelines for compliance to the bioequivalence criteria, if you look at the first column, the Council of the European Community, they are a lot more pragmatic compared to the United States, where if you look at the bottom half of the CMAX requirements, they are much more generous in terms of what can be termed bioequivalent to the innovative product, where you're given a range of between 70 to 143% of the Cmax of the innovator. However, if it's a drug with a narrow therapeutic window, you have to go for a tighter compliance in terms of 8125. In terms of the AUC, for the test product, you have to, however, make 80 to 125% of the reference product. But again, if it's a nano-absorption window drug, you have to comply to a tighter tolerance of 90 to 111%. Whereas FDA allows a ballpark figure of between 80 to 125%, whether it's for Cmax or for AUC. The two main criteria which are used for achieving bioequivalence for the test versus the innovative product. Apart from the bioequivalence studies, there are a number of areas where one has to comply, and these are common to the generic portfolio of products. Whatever criteria is used for the generic guidelines of the United States are the same that is being used for drug delivery systems as well. So you have to be careful about your raw material sourcing, the DMF filing, your location from where you're going to take up your exhibit and your commercial batches, the stability studies that you're going to conduct, certainly on the pivotal batches, the number of batches you do, the batch size, the location, the trade dress. You must have your protocols for both your stability and bioequivalence studies. You must have methods which are validated, and obviously doing all your degradation and impurity profiling, both for the drug and the dosage form. And then I mentioned earlier, your bioequivalence requirement in terms of the reference product. And of course, the facility where you do your studies should be GCP approved facility, and the acceptance criteria that I mentioned earlier should be met. In terms of the capabilities required for developing drug delivery systems are nothing different from the generics or even the new chemical entities, where you need the expertise for both pre-formulation and formulation development, for defining or prototype formulation, to have the desired pharmacokinetic profiles, for which again you need the understanding of how to match the pharmacokinetic profiles for a generic or to achieve a bioavailable product as an innovative product. You obviously need the analytical support in having your analytical profiles, both in terms of the bulk and the dosage form. And more importantly, I think you need a very good patent and regulatory awareness, because patent becomes the key issue in terms of what the patent of the innovator is and how you can infringe, how you can have a non-infringing or preferably a patentable technology, and how you would go about filing your patent and protecting your technology worldwide in order to give you the mileage in terms of extending the patent and the product life cycle. Apart from that, regulatory is quite a complex issue, and therefore your understanding of regulatory is very important. And of course, if you're going to be an NDA or the innovator yourself for your drug delivery systems, you have to go to the clinical studies, and therefore either you outsource that or do it in-house. Your information services for clinical support is also vital. Now there are two options when you have to choose between where you do the development of such a product. Either you can develop your product in-house if you have all the capabilities that I mentioned earlier, or you can go for outsourcing of your technology development. And often it is prudent and it's practical to decide whether you want to outsource or in-house, because in many cases these kind of expertise are available globally, and collaboration often gives you a lower cost of development and time to reach the marketplace, and it has a lot of advantage in terms of the diversity of research, capitalizing on expertise, intellectual wealth, speed to marketplace, as I mentioned earlier, and of course it brings commitment from both sides and gives recognition to both sides as well. Next one. However, you again have two options when you're outsourcing between an academic institution or an industry, because in academia it is probably of a higher risk but at a low cost. It also gives you an access to a total platform technology which becomes probably a property of the industry that would acquire that technology. And it also gives you a worldwide patent rights, and the royalty rates would be cheaper compared to the industry. Future potential is long term. If you look at the industry side, on the other hand, you don't always get the access to the total platform, because that's the intellectual wealth of the innovator drug delivery company. However, they'll develop the product for you, and therefore the patent protection is always product specific. It takes probably lesser time if you go to an expert pharmaceutical drug delivery company, but the cost of the development would be higher compared to an academic institution. The success rate, because of the knowledge, know-how, and the technology availability, the success rate would be higher. You might only get access to worldwide or a country-specific access to the product and to specific markets compared to academic research where you can get global rights on both the technology and the marketplace. The royalty, therefore, there's a price to pay. It takes much, you have to pay a lot more in terms of royalty payments and the years of payment that you have to do. However, future potential is short term because it's only one product specific for which the development will be considered. Therefore, you have to see all the pros and cons before you decide to do your technology development on the idea that you have, whether you develop the product in-house or outsource, and whether you go to academia or industry. You have the option to decide, and often that the academic institution would do the early part of the development in terms of the pre-formulation, the formulation development, and going up to the pilot bioequivalent study. Often with even the drug delivery companies, most of the agreements are such that they develop up to the pilot studies and proving the blood levels both in terms of once a day profile as an NDA or an ANDA product. And after that, the customer as the industry will take up the activities where they would probably prefer to choose to have their own scale-up activities, doing the pivotal studies at their own location for both the pharmaceutical and bioequivalent studies, followed by the regulatory filing, approval, patent control, and product launch. In terms of accessing your technology from an outsourced drug delivery company, this is the list of companies which are working on a large portfolio of drug delivery systems. Many of them have developed very advanced patented platform technologies such as ELAN and ALSA, which are there. And I mentioned ELAN developed the Cardium CD for HMR, and ALSA developed the OROS technology for Pfizer Bacardi XL. Hendrix is the company who developed the bioequivalent generic version of Cardium CD, and there are many more product companies such as Dura, BioVale, Liposome company who are working on amphotericin liposomes for Bristol Myers Squibb and many other companies. And down the line we have companies which are new. Two things are visible here. One, that many of the venture capital companies which were set up in the early 80s are still in the process of developing their platform technologies, and therefore they're still not in the profitable side, but they have got negative growth and running in losses. However, there are companies who have achieved the critical mass of between 500 million to $1 billion company in a very short span of time because of the successes that we mentioned earlier. And I think many of the companies which are probably in the negative today, in view of the platform technologies and the successes they achieve, they probably also will show a much more healthy figure in the future, in the next decade or two. In terms of the technologies that are available, these are some of the known technologies that are available, which any of the companies could access as a platform for developing their ideas in terms of the existing generics or new drug delivery systems. For example, ALSA has the OROS ring cap, which is a capsule with a body and a cap with different dissolution profile and different release profiles with soluble and insoluble coating systems. ELAN has the SODAS and the Pharmazones and the CODAS system. Yago technology, I think we know the geometrics system, which is a 3-layer tablet with the outer and the outer layers being the polymer matrix and the central layer being the drug layer. And the idea is that as it comes in contact with the gastric fluid, the polymers swell from either sides, which creates a porous matrix and also squeezes the central layer, which is your drug layer. And the release takes place from the side of the tablet and also from the porosity created from the swelling and the gelling of the polymers on both the sides of the tablet surface. Timex has recently introduced a Timex technology which is based on gums, and I understand There are a number of products which have been filed with the US FDA with this Timex technology for a number of multinational companies. One or two products have already been approved and many more are in the pipeline for approval. And there are many other technologies such as micropump from Pharmaleaders. These are multi-particulate system, beads in a capsule. Ethical only also has a multi-pore system, which essentially again is a porous beads filled into a capsules. And then we have RP Shearer, who has developed pulsing cap and diff core. Diff core is the laser drill equivalent of the ALSA, where there's a physical hole drilled in the tablet during compression, and that is the area from where the drug gets released through diffusion during the polymer swelling and gelling in the GI medium. And then of course we have Macromed, who have developed the squeeze gel technology, which is the reverse of the ALSA technology, OROS, where the tablet gets squeezed in the stomach, in gastric fluid, and when it goes to the intestine, it swells, and therefore it starts releasing the drug through the porous membrane. These are some of the highlights of technologies that are available, and there are many more that are being developed and being reported in the literature. And I would suggest that people stay in touch with the technologies being developed. Therefore, you don't reinvent the wheel or end up infringing on some of the technologies that are available worldwide. This is an experience that many of the companies face in terms of when they're developing either a generic or innovative drug delivery systems. In terms of the progressive upgradation, I just wanted to give you a milestone in what has happened over the last 2 decades. If you see the bottom column, it says we started with pH-dependent systems, and the two famous products that comes to your mind is a product called Dorix and Eric. Doxycycline and erythromycin were two of the drugs which offered gastric irritation and therefore were not very popular with the doctors or the patients. And therefore Folding Australia developed what was called the pH-dependent beads technology, filled into a capsule, and that went on to become big successes, even overtaking the generic molecule market sales when they were introduced by the multinationals. After that, I think the next introduction were pH-independent systems, and both in terms of matrix, single unit or a multiple unit beads into capsules. The two known examples are Cardizem CD in beads and Procardi XL as a matrix tablet. Then came the modified release, which was a combination of the pH-dependent delayed release or a pH-independent extended release profile that I mentioned earlier. And the famous product of that is Cyclo CD of Eli Lilly, which was a combination of those two and being termed as a modified release product, which is a combination of a pH-dependent and a pH-independent matrix. Today we see a number of work going on in microencapsulation and particle coating. And the famous product that comes to my mind is the Astra product. And I just forgot to mention one more product in the pH-dependent profile many of us are familiar with, is Astra's omeprazole beads technology, which is essentially a pH-dependent multiple unit beads technology. However, Astra has developed now a particle coated product of omeprazole, which is well known by the name of MOPS, multiple unit particle system. And this, they believe, because of direct particle coating, gives you a more reproducible release profiles. It also gives you a better reproducibility in terms of batch-to-batch variability, and it also gives you a better assurance in terms of the bioavailability or the pharmacokinetic profile, because you are virtually coating the primary drug particles rather going through a number of interventions that you require to make a beads product, where you will require to go through mixing, granulation, extrusion, spheronization, drying, sizing, followed by your coating of the beads. And therefore you avoid all those stages of processing when you're going through particle coating. Now this was not possible till about a few years ago, and recently GLAD and Aromatic have introduced a coating technology which allows you to do direct drug layering and therefore allows you to coat the drug particles at its primary stage by eliminating the number of processing that you have to do and also ensuring your batch-to-batch reproducibility in terms of both the solution and bioavailability. And finally, I think we'll see an advent of a number of products coming through the transmucosal route, because if I quote Sir Richard Sykes of Glaxo in one of his recent presentations, he said that the future is going to move away from oral drug delivery systems to transmucosal systems for a number of reasons. One, that it bypasses the first pass metabolism, and therefore all the problems associated with first pass metabolism get bypassed in terms of rapid onset of absorption and action, and therefore you can reduce the potency of the drug and thereby reducing the side effects and the toxic effects associated with oral drug delivery systems. And therefore I believe that that is going to be the next stage where we'll see an explosion of drug delivery systems coming through either the buccal or the inhalation or the nasal route. All three of these, it bypasses the first pass metabolism. And that leads you onto the platform, the basket technology that you require in terms of having that capability and strength to develop yourself as a capability and a strength in the global market for drug delivery systems. Just a few minutes. It really highlights what I mentioned earlier, starting with the Dorix and the ERIC, and then of course the low-sec of Astra, historically going through the progressive upgradation of pH-dependent to pH-independent matrix. Then we have Cardizem CD, and of course the last two slides, just last two products, just shows that particle coating, we only have seen two products purely from the taste masking point of view, but I believe now we have the Astra's product, which is the MOOP system, using the powder and particle coating system, using the same technology used by both Glaxo and Abbott in developing the Ceftin and the Bioxin dry syrup. And the next slide will show you the kind of technology that has improved in the last few years. Today all you need is the backdrop of the white powders that you see and a beaker or a drum full of polymer solution that you need to lay the drug onto the drug during the coating of the weight buildup of the particle size, and then the polymer coating to give you the desired particle size and the control release that you're looking for. This is the last slide, which shows how the technology is moved. If you see the bottom half, the fluid bed technology essentially was available for either fluid bed drawing of granules or coating of tablets. Subsequently, the Worcester technology was developed, whereby you could do a bottom spray coating, and that was the technology used for both the Eric Doric, the Astros technology, and also the Cardizem CD, where they used the Worcester column technology, bottom spray coating, prior to extrusion and spheronization as a technology for making both the delayed and the extended release product. And right at the top, what you see is the tangential spray coater, which is what allows you to do the drug layering with your polymer, and followed by the middle technology, which is the Worcester column, to give you the control release using the polymer system, where there is delayed release, extended release, or a combination of delayed and extended release to give you the desired profile that you want. And this combination of technology gives you the flexibility to offer any kind of control release profile that you wish to develop for your future. Future for thought, just one last slide. I will leave you with two quotes which I think is very apt in today's scenario. One from Darwin, which says that it is not necessarily the strongest or even the most intelligent animal which manages to survive in the process of natural competition and selection, but rather the one which is capable of quickly adapting in the changing circumstances. And finally, David House, the chairman of AAPS, recently quoted that one cannot hope to discover new oceans of opportunity unless he has the courage to lose sight of the shore. Thank you.
Thank you very much.
I'm tempted to ask one question. Madhuri, you asked, you know, a lot of information on the technology used by world over. You give a lot of information on technology that is available the world over. But what about the technology that Brand Taxi used for system?
Well, I guess that technology is available. The one that I don't work for advice.
I don't, but you
That's
guys
why I
want it.
purposely avoided that subject at the topic. Also the fact that it's still under development, and it's not in the marketplace, and therefore it's not right to talk about technology at this point in time. However, the product patented worldwide, and therefore anybody who wishes to know what that technology is all about, is to go to the internet and have an access to the technology that is used by manufacturing for the development of technology.
Thank you, and I think you could be an ambassador somewhere.
And I think in April the publication will be available.
Next year, that was Dr. Khanna Ranbaxy saying that Ranbaxy has the patent.
It has been granted.
It has been granted, so the technology is now freely available to everyone.
The publication will come out
Available for information.
For information.
Not
abroad.
abroad.
Excellent. Thank you very much. Any last question? Big question? Small? I don't know this takes Margaret a few minutes for us to recover from one of your overwhelming powerful presentations. So may I just move on then very quickly to thank Margaret for a marvelous presentation, to show our appreciation by giving him a little award on behalf of the organizer. Thank you very much. Well, this is an honor because Thakur is president of the Indian Pharmaceutical Commerce Association.
From past president,
All right.
the current president to the past president, via the intermediate president.
Thank you very much.
May I now request Mr. Khanna to come over? Mr. Khanna is MSc and PhD, vaccine laboratory. He is also the member of the governing council of Vaccine Science Foundation and member of the board of Vaccine China. He is responsible for the total research activity, and I believe, and there is no question about it, the $60 million product has been under his overall leadership. That was done, and I read on the other day something that Ranbaxy has been given permission to launch one more product in US from 75 milligram every day. Is it right?
That's right.
Which is good, which is making us all feel very nice and proud, and to know that we Indians are not just used to generic products and we have other possibility. Mr. Khanna has been a visiting scientist in Ohio State University, and he has, of course, very well traveled. He's a member of Indian Chemical Society, American Chemical Society, New York Academy of Sciences, Indian Pharmaceutical Association, of course. He has over 29 patents to his credit, which is creditable. And he has about 38 papers presented, speeches delivered. He is a gold medalist. He has got ICMA PCA award twice in 1988 and again in 1992. He has got the DSIR award, DGTD award, FICPK award, whole of them, DSIR award. Thank you very much, Mr. Khanna, for coming here all the way from Delhi. We are very grateful to you for accepting our invitation. And I will request you to chair the next two sessions, which are going to be, one would be Mr. Anil Nimkar on analytical solutions, and other is Mr. Bagade on multi-purpose reaction composition unit. May I request Mr. Anil Nimkar to come over to the stage, please.
Thank you very much, Pharma, Business and Technology, for the invitation to chair the session. I'll just introduce the speaker and then let's spend a few minutes on the topic the speaker has taken for this afternoon. Mr. Anil D. Nemkar, he did his MSc from Institute of Science, Mumbai. He has experience of 20 years in various organizations in the industry. His current responsibilities are sales, marketing, technical support for analytical, life sciences, instrumentation such as FTIR, spectroscopy, UV, etc., etc. He is conducting customer training programs from time to time in various workshops related to the markets and the sales of the instruments which he is selling. He had some training abroad, and he is a member of the advisory committee at Higher and Technical Education. He is also a member of the technical committee of NACON exhibition conducted in Bombay. Well, the subject of his talk is analytical solutions for pharmaceutical scientists. I just expand the subject a little bit, and he will present what he has planned. But analytical solutions are not required for scientists only. In any business, in any industry, we are looking for solutions. And I think that's a very, very important aspect of the business. Since this morning, I think most of the speakers have talked about the problems, difficulties, and the problems they are going to face after the intellectual property is on hold. But in solution, analytical, analyzing the solutions of the problems in business, finance, IPR, is the most important aspect of the business. I hope Mr. Anil will take a few examples which have commercial value for the audience and explain how analytical solutions help the scientists. The other word is the scientists. Well, scientists are the backbone of the industry and R&D. It's the people, the technical people basically they are. Unless we lift them and the industry provides support and educate and bring them up, the industry will stay where it is, and all the presentation which you have seen this morning will remain where they are. Unfortunately, we are in the industry where a lot of investments are required. To bring up the people, industry has to commit certain funds and certain regular investments. If you look at IT, industry is willing to invest 1 to 2% of the investments based on the sales, and that's going to be the investments by the industry to use IT in the industry. But industry is reluctant to spend 2-3% in R&D. There are very few companies those who spend 1 or 2% if you look at it. But the figures of IT is that minimum 3% of the allocation will be required to conduct or use IT application in the industry. Those who are forward-looking companies, I think they have to allocate those funds. But still for 20-30 years, those who are in R&D, they are still reluctant to spend 2% or 3% for R&D. The success of IT had been mainly because there are qualified people in India. A large number of graduates are available. Whereas the pharma industry has not tapped that brain. They are still reluctant to invest on people and make them of that what is required. If you look at any industry, there is a mix of population. You have average people, you have outstanding people, you have good people. But you need few outstanding people to run the show. You don't need all the outstanding people. And you will never have 100% outstanding people in any organization. One has to find some leaders who can drive the company and take everyone forward. And I think scientist word it is used here, that's why I'm expanding. I think it's a very, very appropriate word, and I thought I'll make a few comments. Scientists are key to the success of industry. Essentially, they are technical people. They have to be at par with business and politics. The reason is, today the world is dominated by business and politics. So scientists are suppressed, and they are not at par. And the countries which have allowed scientists to grow and come forward have done very well in science than other countries. With this small few comments, now I'll request Mr. Anil to make his presentation. Thank you.
Introduction. But above all, there are many things which have been discussed since morning, and there are some my present topic which I'm just going to cover, as we have 20 minutes in that. What I just decided to do it because whatever talked about is many problems. And what I'm just thinking to give it the solutions which are available, rather not the solutions in totality, but the tools to get to the solutions. So we'll talk about the tools. So that's my aim, to cover an overview, with which I will take some of the experiments or some of the specific examples at the end, maybe a few examples which have been done very recently. Sorry, this is just to give you all that. Of any pharmaceutical development today. Unfortunately, which I'm just looking for over here right now, many people, nobody is much doing in combinatorial chemistry. I will not be talking much on combinatorial chemistry, but I got a lot of examples on that. But main important thing is the time. Need not to tell about. But this time is again segregated some of the ways which is indicated. What are the market drivers, which again was a talk which earlier indicated, which is in much more detail. But mainly it's reduced time to market, right from the initial stage. Enhanced throughput. You need to have the screen very quickly. You have to do a lot of things in the throughput. Minimize risk of regulatory rejections. Already it was covered. There are possibilities of regulatory rejections which are there. And reduce cost of development, because it's again competitive. So when we think about a drug development process, because most of you are already aware, which is right from discovering test tube to the on the bench scale to the R&D level for development, when you go to the manufacturing, and most important, before reaching to the customers, it's QA and QC. I know that the details will be covered, so already it was done earlier. I'm not going to cover that, but these are the areas. I'm just showing this. I want to show that tools which can be used in these areas, and which are those tools which will be covering one by one. Another very important thing, we all think in terms of cost, or what are the pharmaceutical drivers, pharmaceutical industry driver priorities. And if you consider the cost involved, you can see time to market, the most important part is in discovery, research and development is the main priority. Cost control is only coming when you are talking about manufacturing, little bit in development and QA/QC. In quality, you are bothered at QA/QC. The cost is much more in QA/QC. And in regulatory compliance, you talk about manufacturing and QA/QC. Again, this is going to come in picture of what are the tools which you will cost in various areas. Now, when we talk about pharmaceutical laboratory, it's again the most important is data-intensive industry. Already, Dr. Khanna indicated about IT technology, how it can be utilized. That it needs relevant information that leads to decisions. There is a lot of information with these IT technology, but what information which is relevant and how to come to the relevant information which will come. Better process integration, faster development, less cost, and ultimately successful outcome. Then streamline technology transfer, discovery and development integration, improved candidate selection with lead compounds. Ultimately, today the industry is moving to get much more lead compounds. Now, what are the technologies available in terms of, now I'm talking about these technologies are the analytical technology, all analytical instrumentation available in this process. Molecular spectroscopy, okay, these are the technologies, and where they are used in this process, we can look at UV visible, fluorescence, infrared. Now many times we feel that these are just the traditional technologies, and most of the people are aware, okay, we are doing it for FDA approval, it's required and we want to do it. It's not true. There are many applications which can help you to reduce the time to come to the final lead compounds. Another big thing is separation science, in which GC, LC, GCMS, LCMS or LCMS-MS and electrophoresis, which are very quickly coming up in this area. Another very important area is material characterization. Then after this, you need automation. Many of the things you have to do many times, many of the industries I see 40 HPLCs, 80 HPLCs, 40 spectrophotometers. You need automation in that, as well as robotics, particularly liquid handling, transport, sample preparation, and To have meaningful data, you need to have informatics and collection and retrieval of the integration are used throughout the drug development process. These all technologies are used in the drug development process. Just to mention about very quickly for this technology as an overview, we can go, if somebody wants detail, we can look into that later. We know about UV visible as content in the content determination for uniformity, maybe with the dissolution test, concentration measurements, purity analysis. It is used with the chromophores for identification purpose. When you are having the mixtures together, you can use it for multi-component analysis. Another important area is kinetic studies or enzyme activity, metabolism, and quality control for content uniformity and dissolution. Then next area, again, very few people are using fluorescence spectrophotometry, but which is giving a lot of applications in the area like gene discovery, gene expressions, XTS technologies like high throughput screening, cell function research, functional metabolic or kinetic assays, folding or unfolding of protein, membrane structure studies, therapeutic monitoring, which mainly some unique benefits in drug discovery through R&D is achieved by fluorescence spectrophotometry. This technology is well accepted in the pharma industry, mainly for QA and QC people. It's for biomolecular protein analysis, lead compound identification and optimization. But again, it's a little bit time consuming. There is some other solutions also. Quantitation in biological matrix, compound characterization, formulation development, purity evaluation and stability testing, scale-up and production, process control, bulk drug substance assay. So it's the most prevalent technology used. Today, liquid chromatography is most prevalent technology used in the pharma industry. This is coming up very, very quickly and it's solving many, many problems or giving the, particularly for lead screening and identification, LCMS or LCMS, for structural elucidation, drug metabolism, pharmacokinetics, again people are aware about that, structure profiling, purity and stability, and reaction monitoring to modern drug discovery and development efforts. This technology, infrared spectroscopy, focuses on more information or better information for chemical and physical properties, qualitative and quantitative analysis, active and excipient simultaneously, because we are talking not only just active. You need to know about excipients, extracted mathematically, not physically, because again mathematics, chemometry is widely used in the infrared spectroscopy. And it is again big advantage of this technology which can be used in this area is mainly because it can be applied to solids, powders, granules, paste, suspensions. There is no limit for sample. So it's one of the very important technology for materials characterization and quality assurance need. Another big area which is now again growing up or it's becoming a buzzword is almost 100% analysis. So near-Earth infrared, which reduces the cost of material confirmation and assay controls, is picking up in this area, or rather it's a future for knowing blend homogeneity, placebo tablet assay, contaminant identification, content uniformity, solvent recovery. Many other areas are coming up with FTNR technology. The essence is again time. Analysis time is in few seconds. Another very important technology in mid-ion infrared spectroscopy, as people have heard about combinatorial chemistry, or when we talk about the lead compounds to get by LCMS, MS to get the purity profile. If you want to know whether your reactions are completely formed or whether your reaction is complete or not, you can able to study by infrared spectroscopy on the bead itself. Whether your reaction is complete or not, you can study it on the bead. So you need not to take out, you need not to dissolve the sample in the solution and then put it to the liquid chromatography or LCMS, MS. So you can have the analysis on the bead. So no difficult or time-consuming cleavage, non-invasive, non-destructive. Easy to analyze, linker materials, intermediates, final products, reaction sequence verification, reaction optimization and completion. This is a new technology coming about as a synthesis monitoring systems. So it's a solid phase combinatorial chemistry synthesis which can be achieved by infrared spectroscopy. Okay, there are some of the products in this area. One of the products which I indicated is the usable spectrophotometer. This product is quite important, which I would like to little bit tell about, is solid phase synthesis in combinatorial chemistry. So synthesis monitoring system which is used, or if you want to do the screening, high throughput screening, you can have a microplate reading directly done with the system itself on the plates without doing any changes. Now when we talk about this synthesis monitoring system, you can know this, which saves the time. As I told you, it's on the bed, simply and quickly on the bed. What's done over there, it's a beam condenser, and you're putting your bit directly on the beam condenser, and you take the spectra on it. It's compressed and you get directly the spectra, and you can interpret with the software available with the equipment to know about structure of the compound. Other areas which are for DNA proteins, enzymes, drug candidates, which is for lab. This is for using the spectrophotometer and as an indicator of fluorescence, which is available. Some of the things which I will just go in the area of QA, as we all are aware about QA, usable spectrophotometer. But very few people are using QA, particularly for purity index, a differential scanning calorimetry, which is again coming a field which can tell you about polymorphism, crystallinity, stability, and melting point. This is the area. Calculation of active ingredients and excipients can be done by differential scanning calorimetry. Another big area is residual solvents. Most of the people are using solvents, and in the QA environment, if you want to analyze residual solvent on the drugs or powders, there is no other technology than headspace, which can be combined. Then for nitrogen or protein estimation, you can use elemental analyzer. As far as physical chemical properties, you can use FTR, usable thermal analysis, and HPLC. And I indicated to you earlier, the FTNR technology on systems are available, which can be through the quarantine or in the store, which you can move it and you can take place to place with 2-meter floats, which are shown in this case. Now, reducing the risk of regulatory rejections. The systems now use our ICP technology, inductively coupled plasma, and for drug packaging, the thermal desorber systems, which are used very well in the area of analysis of drug packaging. Not to forget about IQ, PQ, which are the meeting deadlines, which are all available with all these equipments. Another area which is for moisture content and composition analysis is TGA, thermal gravimetric analysis. Then by GC you can have organic colloidal impurities. Then by atomic absorption spectrophotometer you can find out the metal content. This is the area which is in QA/QC. We are already aware much more about the HPLC systems, which give the enhanced performance in active ingredient analysis, impurity determinations, stability studies, and dissolution analysis. Now, just to mention, this is a new technology which about infrared, which I want to just show it. It's just doing the sample as it is without any preparation. I'm just showing one sample for one technology. You can see over here, there is an instrument where you can keep your tablet as it is without any preparation. Suppose even you want to know the spurious drugs, counterfeit investigations. I was hearing this in IPC, people were talking about counterfeit investigation. This is the area where you can put your tablet as it is. And just to show it next, you can see the tablet spectra in one go, and the right side where the spectra which is taken, what is shown in green is how much pressure you are applying it on a tablet, which will be consistently given. So you can able to find out with the technology